Hi, and welcome to 5-Minute Statistics for Clinical Research. My name is Caroline Herborn, and I am part of the Biostatistics team at GCP Service International. It is a common assumption that large amount of data generated in clinical trials can be analyzed in depth, preferably with significant benefits of the investigated treatment or device. In today's video, we will discuss why this assumption holds only for the primary scope of a trial and what is the difference between exploratory and confirmatory statistical analysis. Clinical trials are required to establish two key parameters about a novel treatment or device. They should show that the product is safe and efficacious. This information culminates in the risk-benefit ratio of a product, which is the deciding factor to determine if a product is released to the market or not. Since this is so important, the risks and benefits have to be assessed very carefully and with a high sense of accuracy. While risks are in most cases assessed by looking at negative side effects of the treatment, also called adverse events, the benefit is established by determining the positive impact the treatment has. Ideally, this is done in comparison to another treatment or placebo control. If we assume that, on average, your treatment is better than the comparator, is this sufficient to say that there is benefit over the comparator? To answer this question, a statistical test has to be performed. It basically answers the question how likely it is that a difference could have been observed randomly. The type of test used here has a major implication on the conclusion you can draw, and this is where we come back to exploratory and confirmatory analysis. The main goal of exploratory analysis is to understand the relation of cause and effect, and therefore allow the assessment of relevant influence factors. Based on the outcome of such analysis, new hypotheses can be developed and research questions opened. In exploratory analysis, we can only describe what we see, but we cannot draw any statistical conclusions on why we see it. That means we can say that we observed a difference between our treatment and the comparator, but we cannot say with certainty that this difference is caused by the difference in treatment. Confirmatory analysis, on the other hand, evaluate predetermined relationships with concrete expectations towards the outcome. Hypotheses have to be defined prospectively at the start of the trial and are statistically tested based on predefined models or tests. This gives us the chance to be also able to state that the difference observed are caused by a difference in treatment. The probability of coincidentally observed differences where there are actually none is increasing with every additional test you perform on the same data. This is why you should generally only perform one or very few confirmatory analyses for each trial. An additional analysis should be performed in an exploratory manner to not increase this probability. This also means that you have to be careful with how you interpret exploratory results. They should only be used to generate new hypotheses and research questions for following trials and to assist in interpretation of the confirmatory analysis. But are there no trials that only have exploratory analysis? If you recall, clinical trials are meant to show benefits, but also risks. For drugs, due to the impact they may have on the entire body, this means that researchers have to go through a number of clinical trials before being allowed to release a drug to the market. Initially, this mostly involves few healthy volunteers being treated, and the aim of these trials is to see what the side effects of the drug are. At this stage, since the patients are often not sick and cannot be treated, you do not need to perform confirmatory analysis to show benefit. Similarly, following trials may aim at identifying the correct dose of your drug before trying to show its efficacy in confirmatory trials. So there's a place for both exploratory and confirmatory analysis in clinical research. You just have to be careful to draw the right conclusions from the analysis that you perform. Exploratory analysis can be used to highlight differences, but they cannot confirm that these differences are caused by the treatment. For that, you need to perform a confirmatory analysis. So that is it for today. If there are any additional questions, our team of statisticians is happy to help you out. Leave us a message at statistics@gcp-service.com or leave a comment below. If you are significantly satisfied with the content, make sure to subscribe to not miss the next